Okay, so let's continue with the experiment design and these sample size calculations that we are doing. Now, in the next, in this lecture now, we will explore a few ideas about experiment design, and also we will explore further these false positive, false negatives, ways to reduce the variance of the observations and so on. Okay, so first thing is we have mentioned that when you do many experiments, you have to protect yourself against something called the inflation of the type 1 error. So you will make many more false positives than you think. Let's look at this example. So let's say that you're doing a drug screening experiment in which you will analyze 1,000 compounds. So the reality is that only 10% of these compounds makes a difference or have some effect on the cells, for instance, that you are studying. So, but this you don't know, this 10% you don't know, that is why you're doing your experiment. So in reality, 1,000 out of these 1,000 compounds, only 100 will make a difference, and the other 900 they will not make a difference. But this a state of nature you don't know, because you have a significance level of 5%. So whenever the p-value is below this 5%, you will reject the null hypothesis. So out of these 900 compounds that they are not useful, but because at 5% of them you will think that they are useful. You have been unlucky with your samples, and then you think that the compound is making a difference. So you will have 45 false positives, and in the other 95% you will correctly identify that these compounds are not useful. So you will have this many true negative tests. Okay, that is on one side. On the other side, you have 100 compounds that are really useful, and because you have let's say a statistical power of 80%, you will correctly identify that 80 out of these 100 compounds make a difference. But again, because you have been unlucky with your sample size, with your samples, then you will miss 20 out of these 100 compounds. So you will think that these 20 compounds will not be useful because the p-value was above the level of 5%. So if you count how many errors you have had, okay, so you don't know the true state of nature. All you know is how many positives you have observed. So I have observed these 80 positives, but I have also observed these 45 positives. So how many errors have you committed? So it was 45 were incorrect positives, while this is how many positives you had. So this ratio of how many false positives you have in your positives is 36%. It is not 5%. So you see that by doing multiple tests, we have increased our false positive rate to a higher value than just the 5%. So 5% is good for one experiment, but if you make many experiments, then it will rise, and it will rise it will rise in some amount that depends on what is the true state of nature. So this is more difficult to estimate. All these Bayesian statistics goes into this direction. So it has somehow into account what is the state of nature, and And this 36%, it is called the false discovery rate. Actually, the people working in domains where they use a lot of tests simultaneously, like for instance proteomics, genomics, where they study many proteins at the same time or many genes at the same time, they are aware of this problem and they normally use the false discovery rate. Okay, about variance. Sometimes the experimental conditions induce a variance and we are not aware of that. So here is an example where this person was analyzing the weight of the animals after 18 months, and he was doing experiments in which the number of mice per cage was 1, 2, 4, 8, and then they were measuring the weight of those animals. And there are two important trends here. So one is that the average change when the mice with the number of mice per cage. So when the mice were isolated, so they had more weight than when they were 8. And not only the mean decreased, but also the variance decreased. So the variance when they were isolated was larger than the variance when they were grouped. And somehow the reason could be that when you are alone, you are free to eat as much as you like or as little as you like. But when you are in a group, you tend to behave like the group, and the group also control each other so that they don't allow that any one of the animals eat too much because they also want to eat. So somehow this control affects the mean and affects the variance. So this is just an example about weight, but you can think of whatever experimental variable that you can think of can has an impact on not only the response, so the mean, but also on the variance of your measurements. And this is another interesting idea. So let's we want subjects with few variability. So that is actually why we are using inbred strains in science, because they have very little variability. So this is as opposed to inbred strains, we have also outbred stocks that currently they are not so much used, but there was a period some years ago in which they were more used in science. So these are a couple of examples of these outbred stocks. Okay, so let's look at this experiment. So the point here was they were analyzing what was the time that animals took to recover from anesthesia. So depending, because different strains metabolize the anesthesia in different ways, then they have different recovery times. So let's say that you have number of strains, the mean recovery time for each one of the strains is this one, and also these outbred stocks, you see that they have these recovery times, and these there is a large variability of recovery times. So in some of the groups it is high, in some other group it is really low. The standard deviation within one of the strains is relatively low. We see that the standard deviation here it is 4, 2, 3, while in the outbred stocks it is much more variable. So the average is something, but the standard deviation, there is much more variability around those means. And let's say that we want to detect, so we will do an experiment in which we want to see if by applying some compound, extra compound to the anesthesia, we can change the recovery time. And we want to detect changes of 4 minutes, and this 4 minutes is the gap size, that the effect size that we want to detect. So how many animals do I need to detect this gap of 4 if the standard deviation is this much in each one of the groups? So we already have solved that problem. So we know the gap, we know the standard deviation of the noise, we know the statistical power and the confidence level that we want to have. So we can calculate what is the sample size that we will need for each one of them. So this is the sample size that we need for each one of the groups. In some of the groups that are very little variable, we will need very few animals to detect just this change of 4. In the groups that are more variable, we will need more animals. But in total, if we perform the experiment in these 5 strains, in total we have 79 animals in total. So this is compared to the outbred stocks. Because they are much more variable, we will need many more animals to detect a 4 minutes change in each one of these groups. So we will need almost 200, 300, that is much more than the 80 that we will need with the individual strains. We will come back to this example later after we present the inbred strains and the outbred stocks. So inbred strains are animals that are homozygotic. So that means that given any one of the strains, you will have the same version of the gene in each one of the alleles. So for the gene A, you will have, let's say, a small a. For the gene B, you will have a small b, a small b. So all animals, they have the same version of the gene in both alleles, and all animals are twins of each other. So they have different environments, they have different phenotypes in the sense that the environment has changed the gene expression. So not all animals are exactly the same, but they're twins. So there is no genetic variability, and that is why there is such a small standard deviation. So the standard deviation is not 0 because they're not perfect clones one of each other. Clones in the sense that they have not suffered the same events along their lives and so on. But so there is a standard deviation, but the standard deviation is low because they are all twins. However, when you do an experiment with one single strain, so you are working with this kind of twins. So when you see a difference of 4 minutes between a set of twins and the same set of twins with now a different compound, so how can be sure that this finding of 4 minutes difference can be extrapolated to the whole population? And not only that, but these animals have viability problems. So they reproduce poorly, they are not models for all genetic diseases, so they have experimental problems. And the generalization problem. You have seen a difference between these twins, but how do you know that this difference will extrapolate to other individuals? Okay, so this experimental problems can be a little bit solved by this hybrid F1 homozygotic. So it is you take 2 strains, and each one of the strain have only one version of the genes of each one of the genes, but when you mix them, still all the animals will be the same, but they will have 2 versions of the genes. So again, there is no genetic variability, but they have a little bit more variability of much a little bit more options for choosing the version of the gene to use, and they have gained something that is called hybrid vigor. Okay, but still you are working with twins, so how can you know that your finding in these twins can be extrapolated to the whole population? Outbred stocks can be thought of a community of animals that are they're not exactly the same, so they're not twins, but there is relatively little genetic variability. So, for instance, if you do an experiment in Laponia, this experiment in Laponia, because not many people migrate to Laponia, this community has been relatively close for many years, and the genetic pool, the number of genes that are circulating in the population is relatively low. So again, how can you extrapolate these results on Laponia to the whole world population? So probably they don't extrapolate very well. So it applies to people that are genetically similar to the people in Laponia, but not all people. So now going back to and you would have the mixed stock. The mixed stock is just the whole population of animals. Okay, so going back to our experiment. So our experiment, these are twins. This is Laponia. So you see that if you do an experiment in Laponia, because not all animals are the same, not all individuals are the same, you will need a lot of individuals to make to detect this change. While if you work with twins, this is much easier. But then how can I extrapolate to the whole population? And then here is the trick. So use multiple strains. So if you use multiple strains, you will for instance in humans, if you take Caucasians, so you take Caucasian twins, you take Asian twins, you take Oceanian twins, you take black twins. So if you see this difference of between the control and treatment in all these kind of twins, so it is true that they are twins, and that is easier. That is why we work with twins in the lab, because they are easier to handle, because they have very little variability. But I have observed this change in so many different kind of twins that, okay, they are twins, but I'm more sure that this result will extrapolate to the whole population. So the idea of working with multiple strains is a good one, because they will have low standard deviation, but at the same time, because they are genetically different, they will the result that you have seen in these twins will extrapolate better to the whole population. So this is a very important take-home message. Okay. Still, you can be very careful about trying to control everything. Don't remind this mantra: control what you can, block what you cannot, and randomize the rest. And these people here, they in this work, they were very worried about this reproducibility, and then they did the following. So they were studying different these are different strains of mice, and then they were measuring how these animals behaved. So you can see activity change under cocaine, open horizontal activity. So they were measuring how these animals behaved in different conditions, and they tried to make everything the same except the lab where they did the experiment. So for instance, they were the same research team, the same inbred strains, the same equally calibrated apparatus, equated husbandry, everything that they could think of, they made it the same. The only change was the lab where they did the experiment. So they did this experiment in Portland, in Edmonton, and in Albany. So if you did everything right, you wouldn't expect any significant difference in the three labs. So for instance, for this strain and in this measurement, they see no difference between the three labs, and the same for here. That is what you would expect. But still, you have some responses like this one. So in one of the labs, it was the animals were behaving totally different from the other two labs. So that means that there was still something they didn't control, they didn't know, that was acting differently in the three different labs. So no matter how careful you are in the design of your experiment, there will always be factors that you don't know what they are, but they make a difference. That is why randomization is so important, because if there is, you try to randomize everything that you can think of, because if there is a factor that makes a difference, and this makes a difference is a bias, so it will be factored out, and then it will not affect your results. So as a summary of this part, working with only one strain gives you a lot of internal validity. So that is, there is a high probability of obtaining the right answer. So if the try to get an unbiased result that is powerful enough as to have a low false positive or a false negative rate. And so internal validity is good, so that is what we always should aim in the lab. So we should always try to design experiments that are reproducible, that we get the right answer in each one of them, that we don't have bias, and for the bias remind control what you can, block what you cannot, and randomize the rest. But we are not only worried about reproducing the experiment in our lab, but what we want is that they can be reproduced outside, and at the end, ultimately, they can be applied to a general population that is totally variable. So we want to have external validity. So for that, it is important that you use multiple strains, and also that you make your experiment multiple times. And we have seen this trick that is very useful of making mini experiments as to be able to identify possible biases. Okay, more about blocks. Let's say that we are testing how a new drug affects blood pressure. So you have a group of people. All of these people are hypertensive people, so they have high tension, and we divide these people into two groups of 500. Let's say we have 1,000 people, and then we divide them in 500. One of them will receive the control, the other one will receive the new drug. And then for each one of these people, so we have two groups, group 1, group 2. Within group 1, the first person you will measure what is the blood pressure, and for the second, and for the 500 person, the same in the second group. So each one of these Y's are the response of that, what is the blood pressure of that person. And we have two indexes, one is the group, the other one is the person. So you can compute the average over the people in each one of the groups. So you would have the average of group 1, so this is denoted in this way. So we have the average of group 1, and we don't care the person, so that is the index over which we are averaging, and we will have the average of the second group, and we can average on both groups, so we can compute the average of all people together, so we average over groups and we average over people, so this is the overall mean. And now you can compute how different these means are from the average. So each person from the average of the group, each person from the total average. So the person from the total average, it is this total sum of squares, and because we have 1,000 people and we have to estimate what is the overall mean, we have only 999 degrees of freedom. So every time you make a measurement, you gain a degree of freedom, so you can think of them as coins. So for every new measurement you have a new coin, but then you have to spend these coins in estimating parameters. And for the treatments we have two groups, and again there here a lot of technical details, why number of groups minus 1, but if you have two groups you will need to estimate only one parameter. So once you compute once you estimate the mean of this group, the mean of this other group can be estimated from this one, and the overall mean. The overall mean is important there. But okay, so you need 1 degree of freedom to estimate all this variability. So this variability is the variability from each one, from the mean of this group to the overall mean, and from the mean of this group to the overall mean. But not all people will be equal to their average of their group. So that is the part of the variability that you have not been able to explain. This is sum of the residuals. The residual is what is the variability that I have not been able to explain. So I have been able to explain this much as a function of the treatment, but there is a lot of variability that I have not been able to explain. But the point is that if I had 999 degrees of freedom originally, I have spent 1 for the treatments, and the remaining ones goes for the residual. So also this difference with respect to the average of the group, you have to estimate parameters for those, and then you use these many parameters. Okay, how much variability each one of the parameters explain? So in the treatments, it is you have explained all this variability with only 1 degree of freedom. So this is called the mean squares. So the mean squares is the sum of squares divided by the number of degrees of freedom. So with only 1 parameter, you are explaining 256. While in the residuals, you're explaining a lot, but you have also used a lot of parameters. So each one of these parameters explains 13.61. So when we say I want to detect if there is a change because of the drag, actually what you are doing is comparing this variability to this variability and see if this one is much larger than this one. This is a signal to noise ratio. This is your signal, this is your noise, and then you're dividing the two, and because the treatment explains 19 times more than the noise, then you declare the treatment to be statistically significant above the level of noise. And actually this we explain at some point the critical t value. So when you are comparing the means between two groups, so you have a difference, and this difference you will compare it to the t critical value. If your t is larger than something, you will reject the hypothesis. Here this is an ANOVA analysis, and it is the same. You will compare your statistic. This time the statistic is called the F, that is this ratio between signal and noise, and if it is larger than a critical value, and this critical value depends on the confidence level that you want and the number of degrees of freedom that are involved in the comparison, and it is always, these critical values typically are between 3 and 4, so here we have 3.95. so if 19 is larger than this, then you will say that you will reject the null hypothesis, that is, there is no difference between the two groups. Okay, now men and women, they have different blood pressures. So part of the variability that we were seeing before, part of this variability is caused by the sex. So now if you make blocks and you separate men from women, and men you randomly allocate to control and treatment, and women the same, now you will be able to estimate what is the contribution of the sex. And the contribution of sex, because you have only two sexes, you will also need only 1 degree of freedom. But they explain quite a lot. Actually, in these measurements that we had before, most of the most important part of the variability was sex. It was not the treatment, it was sex. And there is no free lunch. Before we had 998 degrees of freedom for the residuals. Now we have only 997, but still. And the thing is that the total variability has not changed. It is the same. So you see, the total variability is the same. The variability explained by the treatment is the same also. It is the same. But now, all this variability caused by sex has been taken away from the unexplained variance. So that was variability I was not able to explain. But now, a part of it, I have been able to explain it through sex. So when I do this comparison between treatments and residuals, so this comparison now, before I was dividing by 13, you see 13.6, now I'm dividing by 12.13. So before, my treatments were explaining 19 times more than noise. Now they are explaining 21 times more than noise. This is what I meant at the beginning, that blocks help to reduce the variance of your comparisons, because you will compare your treatments to the unexplained variance. If the blocks can explain a big part of the variability, then this variability will be taken away from the comparison, and it will be easier to show that the treatments are effective. And this has to do with the sample size calculation. In the variance that you have to use in the sample size calculation is the part of the variance that is unexplained. So it is this variance here that you have to use for the design of the calculation of what is the sample size that I need. Also, I would like now to introduce something that can help a lot reduce the sample size, and those are factorial designs. So factorial designs are designs in which there are multiple factors that can affect your observations. So for instance, let's say that you have an experiment in which you are interested in seeing what is the effect of some hormone, of water balance hormone, on animals. So you will have two kinds of animals. You will have frogs and toads, and you will have hormone or not hormone, and also because you will measure how much water they absorb when they are given the hormone and when they are not given the hormone, and the amount of water they absorb may depend on whether the previous 2 hours before the experiment they were dry or they were wet. So you will have also the dry wet factor. So one possibility of distributing your animals, let's say that you will have 24 animals in total, and because you have 3 factors, you can make it a design in which you only change one variable at a time. So we have we fix some reference group. So let's say that our reference group is frogs dry with no hormone, and then you change one variable at a time. So you change first the species. Now you change whether they're dry or wet, and then you change the hormone or no hormone. And then in each one of these groups, you put 4 animals. You put 4 animals in group 1. So group 1, group 2. So you have 4 animals in each one of them. Here you have again 4 and 4, 4 and 4. So in total, when you count, you have 24 animals. But now you realize, okay, this reference group, actually I have put a lot of animals because I have put 4, 4, 4, so that makes 12 animals here in this group. So probably this is, I could distribute my animals in a different way. So instead of having 12 in one group that is all the time the same, and 4 only in the others, I could have 6, 6, 6, 6. So I have 4 different groups. I have 1 group here and 3 here. So I have in the 4 groups, I put 6 animals. So still I also have 24 animals in total. So the factorial design, you have 3 different factors. So you have the species, the dry wet factor, and the hormone not hormone. And for each one of them, you have 2 levels. So you have frogs or toads, you have dry or wet, and you have hormone not hormone. So that makes 8 combinations in total. And you can put 3 animals in each one of these groups. And these 3 animals, so you have 8 groups, and 3 times 8, so you have 24 animals in total. So these are 3 different designs. Each one of them is using 24 animals, and not all of them are equally good. So actually the factorial design, this one in which you combine, you make all possible combinations, and you put the same number of animals in each one of them, is much more powerful than these 2 other designs. Because if you count, when you compare frogs with toads, you have in this design, you will have 12 frogs and 12 toads, and you will have 12 dry animals and 12 wet animals, 12 animals with hormone, 12 animals without hormone. Here, for instance, the hormone, not hormone, for that comparison you only have 4 and 4, while here you have 12 and 12. So this factorial design is much more powerful in the sense of a statistical power that we were discussing before. And these factorial designs also allow you to reduce a lot the number of samples. So for instance, let's say that you are interested in the survival of some bacteria under 3 experimental factors. So you have 3 levels of sorbic acid, you have 6 levels of water activity and 3 levels of pH, and then you will measure the density of the optical density that goes through the culture. And because we have 3, 6 and 3 levels, you will have a total of 54 treatments. That is the multiplication of the 3. And we will use a single replicate for each one of the treatments. So for each one of the combinations, we will use only one measurement. And when you compute the ANOVA table, you can count how many degrees of freedom you have for each one of the combinations. So you have 3 water activities, so 6 water activities, so you will have 5 degrees of freedom for that. You have 3 levels of sorbic acid, so you will have 2 degrees of freedom for that, and 2 for the pH, and you have second order interaction. So this water activity with this amount of sorbic acid, it has a positive effect or a negative effect. So these are the second order interactions, and these are the third order interactions. And yeah, we cannot explain everything in just a couple of hours, but there is at the end you have I'm referring to a course that I'm giving on experimental design, and all these details are much more detailed. Okay, so but now the interesting thing is that the third order interactions, we may not estimate them, so that they will remain unexplained, that they will go to the residuals. So we will have 20 degrees of freedom for the residuals with only one measurement per combination. So when you have these experiments where you have many factors, you can make all combinations and make only one single combination measurement for each one of the combinations. You don't have to repeat all these combinations. So that means that you can reduce the number of animals quite a lot. And here is another example. So let's say that you're interested in the biologics bioreactor, and so how cells grow in a bioreactor, and there are 7 variables that you have identified that may have an impact. So the temperature, humidity, pH, concentration of oxygen, concentration of CO2, concentration of glucose, and amino acids. For each one of the factors, you will have 2 levels. So you have a high-low temperature, high-low humidity, high-low pH, and so on. So there are 2 to the 7 possible combinations. But following this approach, where we try all combinations only once, we can reduce the number of samples. So we can reduce. You don't have to make multiple measurements for each one of the combinations. But it is even more. You don't need to try all combinations actually. So with only 64 experiments, still you would have a lot of power to identify the effects of each one of these variables. So you don't need to make all combinations, and this is very surprising idea. So when you have many factors, you don't have to combine to study all possible combinations, and this reduces again the number of animals that you need. So let's leave it here for now. In the following, we will explore some software to make this sample size calculation.